All right, hello everyone. How are we doing today? So this is going to be the exam 2 prep session, little study guide, little review you can go through before the exam. But again, the little asterisks attached to this is not all that's on the exam. You still have to study all the little parts, and always recommend studying the quizzes, and that'll help give you a more rounded picture of each chapter. And then also study the little handouts that we did. They're very important too. So what I'm going to do today is go over the exam. Remember, for the exam, to bring a calculator. Calculator is important. I will give you the genetic code or any important equations that you might need for the exam. So yeah, just make sure you have a scientific calculator. Your phone does not count as a calculator. All right, so we're just going to hop through this all right now, just like I did before, going chapter by chapter and then highlighting any particular area that's important. So the first part here is the linkage and recombination chapter. Don't forget, this is on the exam. So let me zoom into this so we can see it slightly better. And zoom out a bit so it's not in the captions. And there we go. So for this chapter here, remember linkage recombination. The key part to this chapter is understanding what linkage is. So that's when you have 3 groups or 2 or more genes located on the same chromosome. So they're less likely to sort independently from each other. So that changes the typical crossing mechanism that we see in Mendelian genetics. So if you go through this, the key part to this one is understanding how to do these test crosses and to do a 3-point test cross. We'll go over that example a little more later. So right here is just notation for linkage. Make sure you understand that notation. And then incomplete linkage, what it exactly means. So remember that 50% is an independent assortment. Anything less than that means those 2 genes are less likely to sort independently from each other. So that's what this is talking about in this area here. Here's an example of how to calculate that recombination frequency for a simple recombination event. And so this is only 2 genes that are non-recombinants and recombinants. Remember, non-recombinants look like the original parental strains. So here's how you calculate that recombination frequency, where it's the number of recombinants divided by the total offspring. So here, 0.119, then to calculate the percent, you multiply that by 100 for 11.9%. And that also tells you the distance in map units. So you can use that to then map it out. And then this example here, we worked backward. I won't have any direct examples of working backward. That was just to help you guys see that recombination frequency a tad better. We won't have to worry about doing the chi-square test for independence on the exam, just not enough time to get through this, but still understand why it's important, because this is what will help tell you that if we are close to 50%, whether those genes do indeed sort independently or dependently from each other. So again, no direct calculations related to this just because we don't have enough time on the exam. So then, but genetic mapping is important. So this is just showing how we can map different recombination frequencies between 3 different genes here. Again, there are limits to that. And then here is showing a 2-point test cross, which is much simple. But this is the important part, the 3-point test cross. So this is the example we did in class. Make sure you understand this example. And then we did another example in class and on a handout, which I'll show you too. And so just go through these, erase your answers, and see if you can do it all again. I will tell you right now, there's going to be a 3-point test cross that looks just like these on the exam. And you're going to have to map the 3 and state the distances between them. And it's worth, I think, 15 points total, the entire question. So you have to label the non-recombinant strands, you have to label the double crossover strands and the single crossover strands and know which is which. So make sure you look through these different steps and remember how to do it. So for here, you're looking at this, we have the 2 non-recombinant strands right here And then the two non-recombinant strands mean they have the highest numbers and they match parental strands. Then here we have the double crossover strands. They are the fewest in number. So now you want to figure out what's in the middle. So for this one, you look at the parental strands here. They are A, or the non-recombinants, A plus Mg. So here's a plus Mg, so that double crossover event switched A in this one. Here, plus Rg plus, so 2 match. So here is A. So this tells you that A is the one in the middle. So A was swapped by that double crossover event, even though it's not written this way. So that's how you get that. And then, so you write the order then, and then you can calculate the map distances. So looking at the differences between Mg and A, you look at which ones have a single crossover between Mg and A, so that's 15 and 9, and then you add the two double crossovers, so 15 plus 9 plus 1 divided by the total, so 223 for this one then. I think that might not be I think it ended up being 213, or maybe. I have to check my numbers on that. But just make sure you add up the total correctly, multiply by 100, that gives you the percent of recombination. Then with that, you can map the 3. So make sure you can work through all of these 4 steps here and get this map. And then with that, remember we had the interference. Interference is equal to 1 minus the coefficient of coincidence. So if I say the interference is 0.68, you know that 68% of the double crossovers will not be observed. That's the degree of interference in which they have. So you won't have to calculate interference, but understand what this equation means. And if I tell you that interference is something like 0.68, what exactly does that mean? And what exactly does that tell you the coefficient of coincidence is? So you just would plug 0.68 equals 1 minus the coefficient of coincidence, and you can calculate the coefficients of coincidence from that. So then there are also other ways to do gene mapping, such as the somatic cell hybridization, which you make that heterokaryon, and it's a hybrid from the two cells. Some chromosomes get selected and some don't. So if the enzyme is no longer present and that chromosome isn't present in that new heterokaryon, that could tell you that that gene is located on that particular chromosome. All right, so that is genetic mapping. Just make sure you understand that 3-point test cross and how to do it. I'll tell you, it's the first question on the exam. So there's no avoiding it. And then this was the 3-point problem we did in class here. I also put blank copies of this on the files section of the Canvas, so you should be able to access them there. That should be listed under handouts, and I categorized it under exam 2. So this was just another 3-point test cross here, and figuring out the non-recombinants, the recombinants, the double crossovers, and so forth, working out the map units, and then mapping out the distances between each. And then here we also calculated interference with that one. And then we also did that backwards 3-point problem, mainly just showing us up in case you didn't get a chance to finish this, and you could see my answers to this right now. So this one, we worked backwards. If given this, we won't have to do this on the exam. But again, this was a way to check your work and make sure we understand what we're doing, because it allowed us to look at it in a different perspective, backwards. So there are the answers to that one. But we won't go into detail on that now. All right, so that's linkage. Next, we can go to Chapter 9. Chapter 9, I'm at the bottom here, bacterial and viral genetic systems. So in this one, we just talked about the differences between bacteria and viruses. Remember, we know what a virus is, the non-living particle. A bacteriophage is a virus that infects the bacteria. There are the two different cycles. There's the lytic cycle and the lysogenic cycle, so understand the differences between those two cycles. Different types of phages. There's the virulent phages, they're always lytic, or the temperate ones, which can be lysogenic or lytic. And then there are some different types of viruses. They don't all contain DNA. Some are RNA, which are called retroviruses, which mean they have an enzyme called reverse transcriptase, and also integrase, which then can turn that RNA back into DNA and insert it into the host genome. And then here, just, this should be just memory for you guys now, understanding the different genome properties of bacteria versus eukaryotes and so forth. So then here we talked about different forms. Bacteria can gene exchange. So conjugation is via that sex pilus between two bacteria, and then transformation is via the environment. So that transformation experiment that happened, which we'll talk about coming up, and then transduction, which is via a phage. So here it talks about conjugation. You won't have to know the different F factors and things like that. That was just telling you, the nomenclature behind the conjugation event that you could see in the textbook and papers. And then transformation, and then transduction. So again, quite simple chapter there. So then chapters 10 and 11 here, DNA structure and organization. We started out with a little bit of history here. It's important to understand the people who did the key experiments. Just one for you to know, and also might be a bonus question related to the people. So that being said, you don't have to know them, but it could benefit you to know what groups or what individuals did which experiments. So here, Griffiths did the transforming principle, where he transformed the bacteria. Avery, McLeod, McCarty were the one that identified that transforming principle being DNA then. Hershey and Chase had used the T2 phage, and they also determined DNA is a genetic material in the T2 phage. So for viruses, they insert their DNA, and that's what changes the genome of the host. And then Chargaff did the base pairing rules, or A equals T, C equals G, and the ratio of each is consistent between each species. And Franklin and Wilkins did the X-ray crystallography. Remember, Franklin didn't get credit for that crystallography. Watson and Crick then designed the structure of DNA and got that Nobel Prize for that. Then the structure of DNA here. So if you look at a structure, a nucleotide here, make sure you know this is the sugar, or deoxyribose, or ribose. This is the base. You don't have to know whether it's a purine or pyrimidine, but remember the little hint here: if it's two ring structures, it's the shorter word, or purine. If it's one ring structure, it's a pyrimidine. So here, this is the base, this is the sugar, then right here is the phosphate group. So understand the components of a nucleotide structure. That's important too. And then going through here, remember we have phosphodiester linkages that then join these base pairs together. DNA is then forms an alpha helix, and it's also double-stranded. 5' that runs anti-parallel, 5' to 3', 3' to 5'. Remember, between a guanine and a cytosine, we have 3 hydrogen bonds. Between an adenine and a thymine, we have 2 hydrogen bonds. So if an area is very GC rich, that means it's stronger. So if I say something like there are 30% guanines in a sample, there are 30% cytosines, and then you can calculate how many adenines and thymines, since they all should equal 100%. So you add 30 plus 30, 60. Then you take 100 minus 60, which is 40, and 40 divided by 2. So then 20% and 20% for adenine and thymines. So then the different forms of DNA, you don't have to know the direct different forms. Just understand there's a major and minor groove and so forth. But I won't. Make sure you memorize the different forms. Just know that there are different forms. Differences between DNA and RNA. And yeah, that's pretty much it for that. Then here, DNA, remember how it can get packaged in the supercoiling. So in supercoiling, a nucleosome is around 150 base pairs, and it's a little made up of histone proteins. So they're little sets. Each histone, there are 8. It's an octamer. And then, so that nucleosome is at 150 base pairs. And then if you include that histone 1, which is the clamp, it's called a chromatosome. So remember, the center is the centromeres. This is where you get into different parts of that tertiary structure for the packaging. So whether it's heterochromatin or euchromatin. So here, centromeres are a dense region of heterochromatin, that hundreds of thousands of base pairs long. And then telomeres are also heterochromatin, so dense package, and then it's lots and lots of repeats. So down here, so those are called highly repetitive repeats. You can have moderately repetitive repeats or some unique regions, which is where we typically have the coding regions. The telomeres, remember, that's a problem in DNA replication because we don't replicate those ends unless we have an enzyme there called telomerase. I won't ask any direct questions about this, but remember there is genetic information found in the mitochondria and the chloroplast, and they play a role in those species as well. Like all the proteins in the electron transport chain are made by the mitochondria, not by our nuclear DNA. All right, chapter 12 now, we go through DNA replication. Okay, when we get into this, I want to bring up something here that will help you. I put this onto the files section on Canvas. But I'll show it on the screen right now. So this is a little help thing I got put together directly for exam 2. So I wrote as promised. I didn't promise this, but here it is. You should look through this and understand where all these things are and what they are. I know it's a long list, but it's important to understand. So lists of DNA and RNA sequences, RNA molecules, and proteins from chapter 12 through chapter 15, for which you are responsible, and histones from chapter 11. So important DNA sequences to know, what they are, their function, bacterial, eukaryotic, or both. So know which part they are. Are they transcription? Are they translation? Or are they DNA replication? Are they bacteria or eukaryotic or both? So you could make a little list for each of these so you can remember where they are. So like the OREC, how many are in bacteria, how many are in eukaryotic, the licensed origin of replication, which is eukaryotic, the bacterial -35 consensus sequence compared to the Pribnow box, the Tata box, the promoter region, the terminator region, and the core promoter versus regulatory promoter. So make sure you have a little decent understanding of where each of these are found. And then, so these are all DNA sequences here. And they're important for DNA replication. It's just whether you're talking prokaryotic or eukaryotic. And then the RNA sequences here, understand, again, if they are in the post-translational modification step, or they are, not post-translational, post-transcriptional, and then modification, so that's splicing, 5' capping, and that poly A tail. And then if they are just in transcription, so that's where we have like the UTRs on each side, 5' and 3' UTRs, and that +1 start site, which isn't mentioned on this list. But then for translation, then we have that Shine-Dalgarno and Kozak sequence that help tell us where we start. We have codons and anticodons, the protein coding region. Just make sure if you see these words, you understand which part they come from. Then important RNA molecules to know. These are also important as well. So what they are, their function, and if they are in ribonuclear proteins, so meaning they are a combination of RNA and proteins, bacteria, eukaryotic, or both. So this little list here includes messenger RNA, pre-messenger RNA, ribosomal RNA, transfer RNA, SN, which is small nuclear, and this is small nucleolar. These ones are the ones that are important for splicing. And then RNAi, which is interference. So there you have Si and Mi RNAs. So they prevent gene expression. So just know these, and if you see them in a question, you can know what they are then. Then here, important proteins. So if they have an asterisk, they are an enzyme. So imagine if that enzyme or that protein doesn't work, how would that affect transcription or translation or replication? So just know where you find each. And I know it's a long list, but if you break it down, it shouldn't be too bad for you. So like if we don't have single strand binding proteins, the strands will just reanneal with each other in DNA replication. So remember, we open up that replication bubble, these single strand binding proteins come in, they bind, and they protect those hydrogen bonds from reforming. So if they weren't there, the DNA would just come back together. We would never go through replication. If ligase wasn't there, we wouldn't be able to seal the NICs. Yeah. And then here are the different ribonuclear proteins, such as the ribosome, SNRNPs, SNORNPs, and telomerase, which is an important enzymes as well for increasing the telomere lengthening. So it has a sequence of RNA that allows the base pairs to be added for DNA replication. All right, that is that sheet. Remember, that is found in the file section under exam 2. All right, so if we continue back through this now, we can continue on through. Let me zoom in a little bit. Again, here at methylcinostal, they did that semi-conservative model of replication. Remember, it goes 5' to 3'. This is an important figure right here. So be able to fill in the gaps, know where the lagging strand would be, where the leading strand would be, and that it goes 5' to 3' if replication is proceeding in this direction from the replication fork. What an Okazaki fragment is, in that these all start by laying down a short RNA primer right at the start, and then it pushes forward. And then DNA polymerase 1 comes in and removes that RNA primer, and then we keep going. So then this is what it looks like at a replication bubble with the leading versus lagging strands. And there's our little point of origin. So this isn't, we won't have to know as much as this. Again, this is just comparing eukaryotic and so linear DNA versus circular DNA and the different ways it could happen. So here for bacteria, we talked importantly about bacteria here. So the origin replication, that's one origin in bacteria. Helicase then unzips those genes, and single-strand binding proteins bind to prevent that re-annealing. So here are the definitions for each. In helicase, breaks hydrogen bonds. Single-strand binding proteins prevent re-annealing or folding. Gyrase relieves the upstream strain, so it clips it and then puts it back together. And then in the elongation phase, that's where we have primase come in, builds that RNA primer, and then DNA polymerase 3 can then start adding new nucleotides. So remember, different polymerase activity, polymerase 5' to 3' polymerase activity, and then an exonuclease is 3' to 5'. So the difference here then is DNA polymerase 1 comes in and removes those RNA primers and replaces it with those DNA nucleotides, because we don't want RNA in our replicated DNA strand. Then DNA ligase then joins the fragments at the NICs, because we can't form that last phosphodiester bond when we add new primers and add new DNA nucleotides and remove that RNA primer. And then termination phase here, some different proteins involved. And talked about eukaryotic replication. So lysosine factors are all part of eukaryotic for the origin. Remember, eukaryotes have multiple, like hundreds of thousands of origins of where this could start. They have this giant protein complex that comes in to begin replication. Slightly different topoisomerase. In bacteria, it was called gyrase. So different DNA polymerases and no primase. So they are all get like DNA polymerase alpha, delta, epsilon, and so forth. You don't have to super know all the differences there. I mainly focuses on the differences between DNA polymerase 1 and 3, because I know that could get a little out of hand. So then histones, remember, also have to fall off and come back together. Then telomeres at the end, we have that issue, and then telomerase is a way to fix that. So then the homologous recombination, since this isn't fully understood itself, I'm not going to test you, and I know it was also slightly confusing. So just know that this is how crossing over occurs. All right, 13 and 14 here now. So for this one, remember, gene expression is now when we go through DNA, messenger RNA to protein. Talking about RNA here, hopefully everyone knows the RNA structure by now. We have that extra hydroxyl group, so we have a ribose, so phosphate, ribose sugar, and this is actually a uracil base, or it's a pyrimidine. Pyrimidine. There we go. Thank you, captioner. No, that's still not right. All right. Maybe the captioner doesn't like pyrimidine. Nope. Deal with it. All right. So now, remember the differences in prokaryotes and eukaryotes. In bacteria, in prokaryotes, there's no nucleus. So as soon as messenger RNA is made, it could be made into proteins. In eukaryotes, messenger RNA has to get processed and then made into proteins and taken outside the nucleus. So make sure you understand that. Understand the difference between a non-template and a template strand, and how to turn a template strand into a strand of messenger RNA. We did that in class the other day. So here, highlighting the different regions, understand this terminology, the terminator sequence, the coding region, transcription start site, and so forth. And here is where we can see the -35 sequence and -10 sequence in Pribno box and a Tata box. So bacteria versus eukaryotic. And then as we go through here, we make that RNA strand. Bacteria have that holoenzyme. Eukaryotes have that basal transcription apparatus. So here, bacteria have the RNA polymerase and the sigma factor. Eukaryotes have the RNA polymerase 2 and many proteins called transcription factors. So the initiation in each, understand the main proteins involved here. So there's just called RNA polymerase. It doesn't have a number with it for bacteria. Then here, elongation, adding nucleotides, just continues moving down through, and then it hits the termination or the terminator sequence, and then different ways could cause that messenger RNA to fall off, and transcription stops. And then eukaryotes, again, it's in the nucleus, and then it has to undergo processing. So this is where we have the Tata box and some regulatory and enhancing proteins. So initiation, binds, elongation, and termination. Same general process. And then we get that messenger RNA. But remember, messenger RNA in eukaryotes have to be processed. If you see Shine-Delgarno, think prokaryotes. If you see Kozak, think eukaryotes. So the different things that need to be done here, 5' guanine cap needs to be added for ribosome binding, stabilization, and export. Splicing exons together and removing introns. Remember, introns are called intervening sequences. Exons are expressed sequences. This is all done at a spliceosome in small nuclear ribonuclear proteins, or RNPs. So then difference between introns and exons, and then alternative splicing could produce many genes too. And then you can also add a 3' poly A tail. And then remember, transfer RNA carries those amino acids to the ribosome for when we undergo translation, adding them to the 3' end of that transfer RNA. So there are 30 to 40 transfer RNAs that can add an amino acid that can match, that carries an anticodon that matches with the codon on messenger RNA. Then here are the ones that do interference, MI and siRNA. Translation then should be fresh on your minds. Remember, Beadle and Tatum did the bread mold stuff where they discovered the first molecular metabolic pathway, general structure of a protein here. So big part of this is know that N-terminal is made first and C-terminal is the last one made in translation. So if you write out a protein sequence, you always start with the N-terminal, which is the amino side, and end with the C-terminal, which is the carboxylic acid side or the carboxyl side. Genetic code then, understand how to read this table. Very, very important. So AUG is the start codon here. If I say it's prokaryotic, the start codon is slightly different. It's that F methionine, not a regular methionine. So that comes down here when we get to reading the codon then. So then we have the wobble position. So we look at proline here. The last position on that codon could be a UC, A, or G. It doesn't matter. So that's known as the wobble position. Look for that AUG as the start codon. And then remember, there are 3 different stop codons, UAA, UGA, or UAG. So then this is when it puts you in the correct reading frame, and then you match them and make the protein with each amino acid. So yes, you will be expected to do something like this handout here, where you write this sequence and figure out the sequence, the start codon, the stop codon. And so this is prokaryotic. So the first amino acid here is that F-methionine. Then proline, methionine, glutamate, and threonine, if I remember them correctly. So this will most likely be a question on the exam as well with different sequence. So make sure you understand how to do this. Again, these blank handouts have been posted too. And then this one, we did this handout as well. We're not going to do a splicing one, but again, it was matching each one with each other. And I emailed these solutions to you guys too. All right, back to the last little bit here. This is just the different steps of translation. First part includes that transfer RNA charging again, and then different initiations. So with three different sites on a ribosome. We have the A site where it's added, or the aminoacyl site. We have the P site where the peptide bond forms, or the peptidyl holds the growing chain. And then E is the exit site where that transfer RNA leaves. I'm not going to get into too specifics of this, but I enjoyed doing that during lecture. So you don't have to know all these proteins in here. But then elongation takes place, moving down, matching codons to anticodons, growing that peptide chain at the P site, and so forth. Then we hit the termination factors, or the release factors, which comes in. No amino acid, no peptide bond is made, and the whole structure falls apart. And then polyribosomes are where we can have the DNA being directly transcribed by RNA polymerase, and then going directly to messenger RNA. If this was prokaryotes, there'd be no nucleus or processing to go through. So then the ribosomes can come in and make it. So you can have multiple polymerases, multiple ribosomes. So it forms these polyribosomes complexes where you're making lots and lots of protein from one gene sequence. And then also, you don't have to know too much about this, but then those proteins that are made can also be further modified. All right, so yeah, that is this exam. I know I just threw a lot at you, but you can go back through this and pause it and focus on the steps you need to see. And yeah, I do know it's a lot, but you guys should do fine on the exam. Again, it's a bunch of short answers. And then how many? 9 multiple choice on this each one. So not as many multiple choice. And then 5 additional points of bonus are available too. So it's 105 potential points and graded out of 100. But if you listened and understand everything I just said, you should do relatively fine on the exam. All right, if you guys have questions while studying, feel free to email me or ask me on Tuesday if you've watched this by then. All right, hope you all have a great rest of your day, and good luck.